We've got just about one more minute, so welcome all of you here. Just wait a minute or so for everybody else to join the webinar. All right, I think we're ready to go. So thank you all. Welcome to today's Birds of the World Discovery Series webinar, hosted by Cornell Lab of Ornithology here in Ithaca, New York. I'm your host, Laura Kammermeyer, and joining me is Jessica Kane, who is working behind the scenes, helping you with the chat and the Q&A. So today we're going to learn all about red-tailed hawks, which is one of the most widespread and common birds in North America. And I'm going to introduce our special speaker in a moment, but first I'd like to tell you a little bit about the ancestral history of this land. So Jess, do you mind going to the next slide? Great. So Cornell University is located on the traditional homelands of the Gayokono, or the Cayuga Nation. The Gayokono are members of the Haudenosaunee Confederacy, which is an alliance of 6 sovereign nations with historic and contemporary presence on the land. The Confederacy precedes the establishment of Cornell University, New York State, and the United States. We acknowledge the painful history of the Gayokono dispossession and honor the ongoing connection of the Gayokono people, past and present, to these lands and waters. Thank you. All right, so before we get started, just a few tech-related announcements. We are going to be answering the questions that show up in the chat. Into the Q&A. So we won't be looking at the chat so much to answer questions. So if you want to have us consider one of your questions for the speaker later on after, then please put it in the Q&A. We scheduled the webinar for about 60 minutes, but we might go over a little bit, but not too much. Closed captioning is available in English. So if you just want to click the Show CC button, the Show Caption CC, that's how you get closed captioning. The webinar will be recorded and sent out after the event. So it usually takes us anywhere from 1 to 5 days, just depending on how our schedules are. And if you require a certificate of attendance for your college or university, please reach out to us using the contact form at birdsoftheworld.org. So over the past 4 years, the Red-tailed Hawk Project has captured and sampled more than 400 hawks from 7 of the 16 described subspecies and deployed over 100 tracking units. In today's webinar, Bryce Robinson, actually, Bryce, can you put on your video now? There you are. Hi there. So Bryce Robinson is a Ph.D. candidate here at the Department of Ecology and Evolution here at Cornell, and he's going to share what the project has learned from these efforts and what we hope to learn moving forward. So welcome, Bryce. How are you?
Doing fine, thank you.
Great. I want you to know that you have the special distinction of being our most popular webinar speaker yet. There are more than 2,000 people registered for this event, so the pressure's on. No problem, right?
Yeah, no problem.
All right. So tell us more about yourself. What led you to study red-tailed hawks?
Yeah, that's a great question. I've been interested in birds since I was a kid, as a birder, but also, turning into an ornithologist at the end of my my undergraduate degree. As far as red-tails go, I haven't had a lifelong passion for red-tails particularly. I think that came when I started doing field work and started working more with raptors after college. And one job particularly was counting migration at the Go Shoots in Nevada, along Mountain Ridge. And when I was counting the birds that were migrating past me, I started across the season, started recognizing that the red-tails changed in their plumage across the season. And that gave me a really, cool firsthand perspective on how much plumage diversity there is in the species. And as I started trying to learn more about that, I kind of fell into a deep hole about how interesting this species is.
Mm-hmm. That's how it works, doesn't it? The holes get deeper and deeper.
Yeah.
All right. Well, we look forward to your presentation. You can share your screen whenever you're ready.
Okay, thank you.
Mm-hmm.
All right, hopefully you all can see that now. I'd like to thank you all for joining today. I'm gonna give this talk as an introduction to the Red-tailed Hawk Project, for those of you that have never heard of it, and also an overview of why we're doing the work we're doing and why the red-tailed hawk, I think, is an incredibly interesting species. I just want to start with this photo by that my mentor and friend Jerry Ligori took, that kind of captures why I think we're all here paying attention to red-tailed hawks today. They're certainly beautiful birds, and I think most of the people that are tuning in that live in North America see these birds often, and so they're quite relatable because they're often daily parts of our lives if we pay attention to them. But I also, one thing I hope to communicate today for those of you that might think that this bird isn't all that interesting, it's a common partner piece of our lives. I want to convince you all that the red-tailed hawk is incredibly interesting, and I'm going to hopefully go over a bunch of different things about why this is the case, and why we're doing the research we're doing. But I'll touch on a few of those right now. This is a subspecies distribution map that I created for the species, and it shows all of the described subspecies for the red-tailed hawk, and there's 16 that have been described. A subset of those are recognized by most authorities, and these subspecies are based on plumage descriptions across their distribution, and some of those are shown here. But this plumage variation is quite substantial in some cases, and generally matches changes in the landscape across the distribution for the species. So the species is what we call polytypic. And there are many different types across the distribution, and that's a very interesting situation for evolutionary biologists and birders as well. This is another image I like to share that captures a lot of that plumage diversity just in the tail. These are all red-tailed hawks that our working group have captured. And I think it really captures how much differences there are across the distribution in tail coloration, with some birds not even having any red in the tail at all, and also in tail pattern. Another really cool aspect about this species is that its plumage is polymorphic in a portion of its distribution. And for those of you that aren't familiar with this term, it basically means that within a particular population, there are more than one color types that are unrelated to sex or to the age of the bird, and we call these morphs. In the red tail, this looks something like these illustrations here, in that you have a light type and then also a dark type. In the red-tailed hawk, it's very interesting because this plumage polymorphism is only found in a portion of the species distribution. And this begs the question of why we see it here in the western portion, described in the brown, and why we don't see it in populations in the eastern part of North America, like Borealis, or in the Caribbean islands. No one really has a solid understanding of why this is the case, and this is something that I personally am hoping to chase down as part of my dissertation and as part of the work of the Red-tailed Hawk Project. Another important consideration for this species is shown here in this image from eBird Status and Trends. It's the map for the breeding season for the species. And what you see here is a lot of red throughout the range of the species. And that red indicates an overall decrease in the abundance for the species in the breeding season. So although the red-tailed hawk is a common species that we often see perhaps daily, it is on a decline, much like other birds in North America. And so that underscores the importance of understanding as much as we can about the species in case this decline continues, or in case this decline becomes more severe, so that we can enact effective conservation protocols if they become necessary. So now that you know some of the interesting things about, or things that I think are interesting about the species, I want to switch to describing the mission of our working group and what the Red-tailed Hawk Project is all about. Generally, I think our mission can be simply described by this sentence that we're meant to conduct research and also to communicate it broadly. As a working group, yes, we're doing research to understand things about the species, but a very important part of that for me is the communication of that research. And it's not just publishing papers that other researchers can read. It's also communicating what we find out to as many people as are interested. And I think that this photo that my friend Greg took in Idaho really captures what I mean by that. I was out in Idaho catching redtails. Greg was along, and we had caught this bird, and we're working on it, getting all the data we needed from this individual. And we were parked alongside of the road next to a house, and the folks in that house came out to see what we were up to. They saw us holding the bird. Now, these folks, they knew what hawks were, but they didn't know that this was a red-tailed hawk. They had never heard of that species. And so I was able to teach them all that I could while I had that bird about what we were doing and any interesting things about the bird and the species in general. And they were very into it. Those two kids were very into it. And this, I feel like, captures the spirit of the working group. We're trying to learn things and tell as many people that want to know about it, from the general public all the way up to other ornithologists. One very important way we are communicating our research and what we're up to is through our website at redtailedhawkproject.org. For those of you that haven't been to it, please visit the website if you're interested. I just want to show you some of the things that we have on the website that I think are important. One of which is the blog, and the blog is basically a newsreel for our working group. We share things that are, recent findings. Say if we publish a paper, it'll show up on the blog, but we also share interesting stories about the work we're doing as we're doing it. For instance, some of the results from tracking efforts that we have, or just interesting tracking stories from the birds that we have tagged. We also have a description of the main focus of our research, that being, any of the projects we have going on, but also there's a description of who's involved, which I think is important for people to know. So you can follow that tab and go to the movement ecology section, for instance, and see what it is what type of questions we're trying to answer with our work in the movement realm. And also what I'd like to build is profiles for all the individuals that we have tagged and all the information we gathered for every individual. Another thing that I'm currently building on the website is the subspecies, is under the subspecies tab. And if you follow that through, you'll go to what is a subspecies guide. And you can see here that at the header is a distribution map that I showed you just a few minutes ago. But underneath that distribution map is a list of all the recognized subspecies, or at least all of the described subspecies. And what I'm building is a guide or a description of the identification aspects of all of this subspecies. So what features about each type do you need to know to tell them apart? And then once there's that description, we also have a link there to each subspecies in the Macaulay library if they have a link to that subspecies for the Macaulay library, so that people can learn from the infographic how to tell these subspecies apart, and then directly go into a photo pool and see if they can see what's described in the infographic in real photos. Our mission can be broken down into three facets that I think are important to touch upon. The first being to promote collaboration. What I hope that our working group shows is how powerful collaboration between researchers and many different stakeholders can be in answering or in understanding a species and furthering our understanding and conducting research. One thing that we're doing is trying to involve as many researchers as are interested in our project to help build these large data sets that we can use to have more power to answer the questions that we have about the species, ecology, and evolution. So you can see here, this is an an overview of all of the institutions that are contributing data to our working group. And you can also consider that each of these institutions is essentially a touch point for anybody outside of the working group that wants to understand anything about the working group. So it's a strong base network for a broader network of people interested in the red-tailed hawk. What I hope is that we can provide a network of people who are interested in the species in any way, such as wildlife rehabilitators who work with the species in their rehabilitation effort, veterinarians, birders, and also falconers. Anybody who's interested in understanding or learning more about the species or contributing information. I'm hoping that this working group provides a network for these people to communicate and share information with each other. Another thing we're hoping to do is to fill knowledge gaps about the species. And again, considering the downward trend in the breeding season for this species, these knowledge gaps are important to fill because we need to have a full understanding of the ecology and the evolution of the species if we're hoping to enact effective conservation protocols. I think a lot of folks would consider the red-tailed hawk a common species. And so it might be surprising that we lack an understanding about many different basic things about the species. And I've created this word cloud here. It's not so important to read all the different topics that we lack an understanding, a full understanding for the species, but just understand that there's a lot of fairly basic things that we still don't know about the species that we as a working group are trying to answer. And the last thing I think that's an important aspect of our mission is to develop the species as a model system. And many people, I think, are familiar with the term model system, but if you're not, a good example of a model system is mice that are used in cancer research. In birds, model systems are things like the chicken or the zebrafinch, things that are easily kept in captivity that we can do experiments on. So the red-tailed hawk, I think, can serve as something of a model system for raptor ecology and evolution. But I want to explain exactly what I mean or what I think it can provide for us. I think because the red-tailed hawk is a common and widespread species that is relatively easy to work with, it's fairly easy to capture, it's a hardy bird, we can use it to do research that will answer questions about the ecology and evolution of the species that might provide broad insights into raptors in general. We can also, through our work, because the species is relatively easy to work with, we can test methods and protocols that might be, and learn some lessons on a more forgiving species that we can apply to species that are riskier to work with or harder to work with, so that we can make sure that those methods and protocols work when we apply them to species that we don't get in our hands more often or as often. And then again, I'm hoping that our working group and our work with this species can develop a model for effective collaboration between anyone who's interested in learning more or conducting research on this species. And then finally, I want to just touch on how I think that our working group can provide, or by filling knowledge gaps, can provide information that will really bring the excellent birds of the world species account to the next level. And that's through publishing papers that will fill knowledge gaps that are outlined in the species account. But I think there's also an opportunity here to figure out how we can build large data sets that can actually be piped into the birds of the world account and provide some actual data-driven aspects to the birds of the world account, much like eBird data is providing new insights into the distribution of the species, and especially things like the status and trends map. We can do that with other data. Now that you know what we're doing and the general mission behind our project, I want to show you, or I want to explain exactly how we're going about doing this work. And it's all based on this idea of the extended specimen. I think there's probably many of you out there that have heard of the idea of the extended specimen, but for those of you that haven't, it comes from the museum world, where you have a voucher specimen, which is generally a round skin that's housed in a drawer. And that voucher specimen provides an overview of the plumage of that individual. Oftentimes it's matched with a tissue sample that's in a freezer, or there's tissue on the toe or fleshy parts that can be used for genetic analyses. On the tag or in the database for that museum, there are a bunch of different information that the collector noted when they collected this bird. So that extends the utility of the specimen with all of the data that's attached to it. For our project, we're using that extended specimen in museums, but also we're adding this digital specimen idea through standardized photographs that can act as a digital representation of the plumage of every individual. And this digital specimen will have a bunch of data attached to it that will extend its utility in the future. The standardized photos that we take are taken against a neutral background that you can see here. This neutral background is meant to simplify and capture the plumage of the bird. But we also take these photos with a color standard that you can see in the bottom right here, so that we can color correct every image so they're more appropriately compared with one another for every bird that we capture. And we can also use that color standard to extract coloration data from the image. So it's a very important part of the digital images. We take, this is an example of the suite of photos that we take. We take these for every single individual, and it's meant to comprehensively describe the plumage for the individual. But you can notice that we're likely getting a bunch of other information from these photographs, like the color of the fleshy parts, any molt information, things like that. And along with all the morphometric data, the blood sample we take, if we're putting a transmitter on the bird, we're getting all this information from every individual we handle that turns into an extended amount of information that can be pooled together to create these large data sets that are centered on every individual, and that we as researchers can share, if it's standardized, among each other and increase our ability to answer the questions we're after. So we're building these data sets, and I just want to transition into sharing what some of these data sets can answer for us and how we're using those in our research. We are tagging a lot of individuals with transmitters to understand where these birds are moving, when they're moving, and hopefully why they move, or why they move in the way they do. And we do this through attaching these transmitters on the back of the birds. They're solar GPS units. They're attached on a backpack style harness that you can see here on this bird that Nick is holding. The units that we use for our working group are what are called GSM units. They are GPS transmitters. So they take a GPS location as the bird moves around the landscape, and they store those GPS locations on the transmitter, and then they function through the cellular network. So you can think of it as working the same way that your cell phone does. Anytime that this unit is in service, it's able to, on a schedule, offload the locations that it's taken. We program our units to take locations every 3 hours and then offload those locations once a day. So as long as it's in cell range, it will transmit those data through the cellular network, and we'll receive those on our computer and even on our phones, so we can see where that bird has been for that day. But we're tracking a lot of birds that might be out of cell range, especially when they move north into places like Alaska and Canada for the summer. So we often don't hear from these individuals for much of the year. When they return back into cell service for the next winter, that's when we get the data. So I just want to turn to this image of Nick because I want to touch upon some of the work that Nick is doing for his dissertation in Michigan, because it's a good example of some of the things that we can use these movement data for in our project. Nick is looking at the movement of red-tailed hawks in the Great Lakes region. For those of you that don't know, red-tailed hawks don't like to fly over water, so as they move north during spring migration, the lakes segregate birds along their shorelines as they move north. And this spot here in northern Michigan at the Mackinac Straits is a place where red-tails group together in high densities as they come north, because they need to cross this 5-mile stretch of water here. And they basically wait to do that until conditions are right. And so at some points in time, there's hundreds of red tails in the air, and it's quite a sight. And Nick's really interested in understanding this behavior of crossing the straits. What factors influence the decision for when a bird is going to cross the strait? So you can see this individual here. It mills about for quite a while until something changes and it makes the decision to cross. And Nick is interested in understanding what's behind that. And that's one part of the dissertation that he is working on at Michigan State. And stay tuned for more of what Nick learns from migratory red-tailed hawks in this portion of their range. Other things that we are learning about the species that we tag are more basic. Here's a harlani that Jesse tagged in Salt Lake City. And we have a full cycle picture of where it moves throughout the year from its breeding in a breeding area in Alaska to its wintering area in Utah. We can see that it has a fairly consistent routing along the Front Range of the Rockies going north, both in spring and fall. And that's something that we can gather for every individual that we tag and answer any number of questions. So here are two individuals, two harlani that Neil tagged in northern Idaho. Both of these individuals, although they're harlans, ended up breeding in very different locations, one in central British Columbia, the other in western Alaska. So although they're wintering in the same area, they come from very different breeding locations. We can answer, how often does that occur? Does it occur on the other end? Do birds that breed in the same area go to very different places to winter? Why that might be the case. But we can also see here a very cool difference in the fidelity to their route between seasons. So Hobbs on the left here took very different routes between spring and summer. Is this something that this bird does every year? And what's behind that decision to go through central British Columbia in spring and then return along the Front Range in fall? Calvin, on the other hand, seems to have a very tight movement, a very similar movement between spring and summer. So we're interested in learning more about what's behind this. Do these birds take the same path between spring and summer, and do they do this every year? And we can answer this question for every bird that we put a tag on and get a distribution-wide understanding of this. Another thing that we can answer through our tracking data is we can understand more about the distribution of plumage polymorphism and what might be behind that distribution. And actually, plumage polymorphism and tracking is why the Red-tailed Hawk Project started in the first place. And so I want to describe that work that I started with my colleagues in Kansas. My friend Luke and I started talking about the birds in Kansas. Mark Robbins and he had been discussing all the dark types that they saw during the winter in Kansas. Mark and other birders there had been interested in knowing where these birds originated for a long time. And you can see here that Kansas, the place I'm talking about in eastern Kansas, is very far east from the breeding distribution of polymorphism, or what we understand as the breeding distribution. And it begs the question if these birds that are wintering in these areas are coming from directly north in parts of Canada where we didn't realize that dark types occurred previously. So we were interested in putting transmitters on these dark types in Kansas and seeing where they spend the summer or where they go to breed. And we found out that all of these birds that we had tagged in eastern Kansas were taking a northwestern trajectory in the spring and ending up in the northwestern part of the continent, well within the region or within the distribution of what we currently understood or understand is a distribution of polymorphism. So that gave us a really cool insight into where all these dark types were originating. But those of you that pay attention to red tails in the east might know that every year there are dark red tails that show up during migration and also during the winter in eastern North America, particularly in the Northeast. And so this question of where the birds in the Great Plains, although we answered that question, we still have this question about where these birds in the far Northeast originate. You can see here from the distribution or from eBird data for Calurus abiaticola tag, which is generally just the dark types. You can see that this happens across the Northeast in very few numbers, but it happens every year. Birders have been interested in this question for a long time, and as a working group, we banded together to try to figure out where these birds come from. The idea was either that they come from directly north, or they are the very eastern extent of that same pattern that we saw from the birds that are wintering in the Great Plains. That seems quite surprising if this were the case, but it could be the case. So we weren't convinced that they were coming from directly north, so we decided to tag them. I won't go into the details about how we captured these individuals, but I feel quite lucky that we were able to catch 4 in all of our efforts. And I think that these 4 individuals also underscore the importance for our working group in that it is a collaborative effort between multiple institutions. And it's the only way we were going to answer this question in a more robust way. So I'm really happy that we banded together to answer this question. What we found out is that these birds do migrate north to breed or to spend the summer well within the distribution of the subspecies abiaticola. So in this picture, or in this image, you can see the routes of all these dark types that we tagged and where they ended up for the summer. All the light circles are light morph individuals that we had tagged where they spend the summer. So you can see they're spending the summer in similar areas in eastern Canada. But the question still remains of whether or not they actually bred. And so I want to share some images that help us understand what they were, what their behaviors were like during the summer. And so I want to just quickly break down this image because for those of you that aren't familiar with looking at home range maps, I think it's important to show what this actually means. This in this picture and in future pictures, this number and letter indicate the age of the bird. And so that says that that bird's a second cycle, but it basically means that it's in its second year of life and that that bird is a male. We had these birds sexed molecularly, and so we know for sure that this was a male. In this contour line here, this indicates that the bird spends 95% of its time within this line. So during the time bin that I've binned for this bird, which represents the breeding season, the bird is spending 95% of its time in this range. The darker colors indicate areas of high use. So this is where the bird is spending a lot of its time. And then this number here is the actual area that the bird is spending 95% of its time. And you'll see that this bird is spending 95% of its time in a 9,000 or in about a 10,000 square kilometer area. That's very huge. Compared to another second cycle young bird in our data set, there's a similar massive home range during the summer months for these species or for these individuals. So we actually know, or we expect that these young birds in their second year of life shouldn't be breeding. So we did kind of expect to see this massive home range, but this light bird in particular is almost 100,000 square kilometers of a home range. That's massive. It's very exciting to see. The adults had a much smaller home range size. And you can see here that the males have a fairly consistent home range size between the two that is somewhat larger, but the females have very small home ranges that have that tiny dot of use there in the middle. And we think this is a reasonable representation of the incubation period for these birds. So these birds were incubating and brooding nestlings during this time, as told by that centrally located tiny home range. The real takeaway here is that the morphs don't seem to differ. So the dark types, we think it's reasonable to assume that the dark types are breeding in this area, and that polymorphism probably extends throughout the Canadian boreal. And this is really important if we are to answer questions about why we see plumage polymorphism in some parts of the range and not in others. It's really important and foundational, in fact, to understand where it actually is. And so we've made a step forward in refining our understanding of polymorphism for the species so that we can now continue research. Another really cool movement story I want to tell is about this bird that Neil tagged in northern Idaho. This bird is a second cycle. It has it has some Harlan-eye features, but it has a fully red tail. And in its in the first year of migration, or in the first winter when Neil tagged it, it spent its winter here in Idaho, or at least from when Neil tagged it to when it started migrating north. It migrated north and spent the summer on the Kenai Peninsula near Homer, Alaska, which was really fun to see because it took this coastal route, island hopping along the islands of British Columbia and Southeast Alaska. This is the first red tail in our set that we had make this migratory movement. But talking to people who live in that area and pay attention to red tails, we know that red tails do move through there during migration, but it was really exciting to capture that. In the fall, the bird moved south on a totally different route, taking the Front Range and moving down to spend its winter in Montana, and even moving into Wyoming for a time. And this was really exciting to see it have this very different winter location between the two winters. The next spring, it did indeed go back to that spot on the Kenai Peninsula. We know from the breeding movements during the summer months that it didn't breed in that first summer that it spent up there. But the really exciting thing is that this winter, it has stayed up on the Kenai Peninsula and is spending its winter along the highway there just outside of Homer. I think that that's very exciting. It's very perplexing. So we want to know how common is this? How often do these young birds spend wildly different their winters in wildly different areas? Is this difference between breeding seasons or between wintering locations, is it common in the early parts of individuals' life, or did we just catch one bird who did a very unique thing? The other interesting thing here is that although there is documentation of red tails spending the winter in Alaska, it's very uncommon, and it's cool to see one of our birds that was captured during the winter in the lower 48 actually spend a winter in Alaska. So now I'd like to shift into the evolutionary genomics aspect of our research. This is what I'm focused on primarily for my dissertation work at Cornell University and with the Cornell Lab of Ornithology. But before I get into it, I want to make sure that everybody knows what I mean when I say the word genomics. I think most people are familiar with the word genetics. Genetics generally means information from one or few genes on the genome. So that's a snapshot of one or a few locations that is basically the tip of the iceberg when it comes to the genome, but it provides us information that we can use to understand the relationships between individuals. Genomics is like the big data for genetics. It is information gathered from across the entire genome, or a good representation of the entire genome. And it's a lot of data. So it's a lot of information that we can gather from the genome. Phylogenomics is basically using that genetic data to understand relationships and the evolutionary history of individuals that you can include in your research. For me, I'm interested in understanding all the relationships and the evolutionary history for all the diversity we see in the species. So how related are all the different subspecies? One question we can answer from this work is that we can gain an insight into whether or not plumage is a good indicator of how related certain subspecies are. So there are certain subspecies that look more similar than others. And then there are subspecies like birds like Costarricensis that occurs in Costa Rica, that middle orange bird, or Harlani that are very different in plumage compared to the rest of red-tailed hawks. And we can use genomic tools to understand if that's a good indicator of how distinct they are, or how unrelated they are to other red-tailed hawks. One question that people have had for a very long time, and there's been a lot of debate about, is whether or not Harlani, the subspecies Harlani, should actually be considered its own species. And for those of you that don't know, Harlani that breeds in the Northwest in places like Alaska, British Columbia, and the Yukon are extremely different in their plumage compared to other red-tailed hawks. So much so, in fact, that a lot of individuals lack red they lack red in the tail entirely. There are very strange-looking red-tailed hawks, and people have argued that because of this, that represents how evolutionarily distinct they are from the rest of the red-tailed hawks, and that they should be considered their own species. We can use genomic tools to figure out whether or not that is the case. Another really cool and untouched aspect of the relationships between the red-tailed hawks is the rufous-tailed hawks. Rufous, for those of you that don't know the rufous-tailed hawk, it is a red-tailed hawk that occurs on the southern tip of South America in Argentina and Chile. They are surprisingly similar, given that their distribution is very separated from the rest of the red-tailed hawks. They look very similar to a red-tailed hawk. And I bet if you showed a birder in North America a rufous-tailed hawk and asked what subspecies it is, they would probably tell you an answer and not tell you that it's a rufous-tailed hawk. They're very, very similar. There's been some previous work into the relationship of this species to the rest of the red-tailed hawks, and it's actually quite interesting. I've shown there the phylogeny from Rising. It's a portion of the overall Buteonian phylogeny, but it shows the relationship of rufous-tailed hawk, or Buteo ventralis, with other red-tailed hawks, showing that it is more similar to Calurus than Calurus is to Borealis, indicating that genetically it is very similar to other red-tailed hawks. I'm including this in the phylogeny that I'm putting together for the species, so we'll actually know how similar it is to the entire, or to all of the subspecies of the red-tailed hawk. The other question we can answer is, how have islands shaped diversity in the red-tailed hawk? So there are a few subspecies that occur on islands off the coast of Mexico and also in the Caribbean. And for those of you that don't know, islands, isolation can generate diversity in species. So if things are separated geographically and they don't share their genes, over time they can develop differences that might turn in and eventually cause them to turn into different species. Islands have a big role in this, and so we can look for the red-tailed hawk, something that doesn't like to fly over water. Have islands generated a lot of differences for these individuals that are occurring on the islands compared to the continental subspecies? So that's something we're looking into as well. The way we're doing this is from samples of every single described subspecies for the red-tailed hawk. So we have built a sample set of 77 individuals that are from all of the subspecies, including the rufous-tailed hawk. And what we've put together are a combination of blood samples from our work across the distribution in North America and in the US and Canada, combined with topaz samples from institutions or museums across North America that you see listed here in the circle. And I just want to say that this phylogeny that we're putting together would not be possible without the help of all of these specimens from these institutions. And I really want to thank all the collections managers and curators for their willingness to share toepads that are going to allow us to understand the evolutionary history for this species. Before I dug into sequencing all of the different subspecies, I wanted to make sure that the approach that we were going to take to this would work. And I'm not going to go into any of the details. I just want to show you some of the results from that effort. But we did a pilot run involving 4 of the subspecies to see if the combination of the 2 different tissue types would work and if we could get whole genome sequences for these birds and create a really robust phylogeny. And I'll just show you the results, glassing over a lot of details. But here are the results from those samples, and showing that the birds are grouping in a fairly predictable manner. They group by subspecies, as indicated by the colors. And you can see the relationships of all these individuals. And it's very exciting. So we were quite successful in our attempt to get whole genome sequences from these individuals. And so we've moved forward in our attempt to build a comprehensive phylogeny for the species. And there should be results from that coming fairly soon, actually. So stay tuned for that. One thing I want to touch upon in sharing this is that the subspecies harlani here is nested well within the other red-tailed hawks, indicating that it is quite similar genomically to the rest of the red-tailed hawks. And this makes sense because anecdotally, we know that they interbreed. There's a lot of individuals out there on the landscape that look like intergrades between other red-tailed hawk taxa and harlani. And then we have direct observations of mixed pairs and nests. So we know there's a lot of gene flow between these. But I think this helps support that we should consider the harlani part of the red-tailed hawks. Now I want to touch on a few avenues of research that we're shifting into that have come as a result of the work from all the birds we're capturing, and something that we didn't really plan on focusing in the first place, but that as we gather more samples, we realize that there's an opportunity to conduct research to understand some other aspects of the red-tailed hawk. And one of those is plumage aberrations. In the red-tailed hawk, there seems to be a lot of birds that have these plumage aberrations that look like this bird here in the photo. It seems to be fairly common and also seems to be more common in red-tails than other raptors. We want to first know if that's the case. Is that true? And if it is true, if there's an explanation for that. We also are interested in figuring out what different types of plumage aberrations occur in the species, and is there one type that occurs more often than others? Things like albinism or leucism, and those types of things. And then another really important aspect of plumage aberrations is understanding how they actually impact the bird, or do they impact the bird? A lot of people, I've heard people say that, if you have a bird with a plumage aberration, it's not a good thing. An all-white red-tailed hawk is a problem for that red-tailed hawk because it sticks out like a sore thumb, for instance. A lot of people say those things, but I don't think that there's any data that really supports that. And so we're interested in figuring out how do these plumage aberrations actually impact these birds. And we recently had in a trip involving some Cornell undergrads, myself and some key red-tailed hawk project participants, we had some luck in Oklahoma when we were trapping, in that we captured this beautiful all-white red-tailed hawk. You can see Nicole here. She was very excited holding this glowing white red-tailed hawk. This is the first all-white red-tailed hawk we as a group have captured, so we were quite excited about it. And you can see here that the bird is almost entirely white, but it has pigment in its eye and in its bill. It also has some pigment there on the back of its head and in some parts of the face. So there is some pigment in there, which is very interesting. It also seems to have reduced amounts of pigment in the talons, so it's impacted the talons as well. So it's a very interesting and fun individual. And although we were there to put transmitters on birds that will be used in my dissertation work, I was at first reluctant to put a transmitter on this bird, but after some discussion with the students and some pressure, I gave in, and we decided to put a transmitter on this bird, and so that we can track it and see where it goes to determine whether or not it's a migrant, and follow it over the course of its life for as long as the transmitter lasts, which is really exciting. And I'm looking forward to finding more out about this individual. So I just want to share with you what we found out about the movements of this individual since we tagged it. And basically this image right here shows you what we found out. And it's basically nothing, because the transmitter, because it's a GSM, hasn't sent us any data. And I think that's because this area that we tagged it in is a 4G dead spot. There's no towers near there, so the bird is not in cell range, so it can't give us data. And that's fine for a migrant individual that will soon start migrating and find its way into cell range and give us the data from the time that it was outside of cell range. But after I shared this bird on social media, I was contacted by this gentleman, Phil, who claimed to know the red tail that we had captured. And he told me that he has been watching that red tail for about the last 8 years, and that it is actually a resident breeder. And after talking more with Phil, he shared some photos and told me about, what he knew about the bird and where the bird that he was watching is located. And I think it's pretty certain that this is indeed the same bird. So hopefully that bird at some point over the course of the season, even though it breeds in that area, it will find its way into the cellular network and give us the data. But we at least know where it hangs out. And some other interesting things that I learned from Phil is that every year he's been following this bird and photographing it. And this is a photo from a few years previously. And you can see here that this individual actually, in previous years, had more pigmentation in its feather than it does now. You can see in the tail and in parts of the wing and in parts of the body, it actually had flecks of color. And this is a really exciting insight into this individual, and the idea that the amount of pigmentation in the bird might change over the course of its lifetime, whether it's a reduction as the birds age, or perhaps it changes one way or the other, more pigment or less pigment with each successive molt. We don't know this, but from this individual, we might be able to figure that out. By now, we've caught a lot of red tails. I think Laura mentioned we were in the beginning that we're over 400 in our data set. So by happenstance, we have actually caught a number of individuals that have plumage aberrations, like this one here that Neil caught in Wisconsin. So we're starting to get samples, blood samples, and images from all few birds that are starting to build a data set of plumage aberrations for the species, and I'm really excited about that. The last research avenue that I'd like to touch on is hybridization. With eBird and Macaulay Library, we're starting to build a better understanding of how often red tails hybridize with other species. We know that it happens with red-shouldered hawks. We know that it has happened in a few instances with Ferrugin's hawk, and we also know that it happens with rough-legged hawk. What we'd like to try and put together is a perspective on how often it occurs, and also start to catalog with which species it occurs, and perhaps which species does occur more often than others, and what factors might be behind that. For the rough-legged hawk, I think there's a very interesting story happening with the rough-legged hawk, because the more people that are out there on the landscaping taking photos of hawks and sharing them in the Macaulay Library through their eBird checklists, we're starting to understand that hybridization with rough-legged hawks happens more than we originally thought. And there seems to be birds popping up at least one a year that are new. And we, as a group, actually ended up catching a hybrid red-tailed hawk, rough-legged hawk in Kansas. My colleague Luke and I caught this bird, this juvenile bird. So we're starting to get some samples from actual hybrids. And so we'll be able to do some genomic work to figure out whether or not answer questions about whether or not the plumage is a good indication of their parentage. So are things with a more half and half mix, are they, first generation hybrids? Are things that are more on the rough-legged hawk spectrum, more towards the rough-legged hawk? Are they, things that where hybridization is one of, a few generations back and they've since back crossed into the rough-legged hawks, things like that. We can also I should also say that Rob Dominich out in Montana has caught 2 hybrids now, and we actually have the blood from those as well. So we're starting to gather a data set so we can answer questions about how hybridization might impact each species, and what things might, how does hybridization, for instance, impact the plumages we see in particular parts of the distribution in Harlanite. So that's something that we're currently working on, and I'm really excited about. So I'd like to wrap up with Jerry's photo again. And hopefully I've convinced you all that the red-tailed hawk, even though it's common and we see it often throughout the landscape, it is an incredibly interesting species, and we are working together to try and understand more about it. With that, I just want to say thank you all for joining and listening. I've included here QR codes for our website in case you didn't write that down earlier in the talk, and also a QR code for our Facebook page, which generally what we share on our Facebook page is it matches what is shared on the blog. I also want to say anybody who's interested in this work, you feel free to reach out to me. I've included my email there. And I also want to say thank you to everybody who's contributed, whether it's data, money, their time. This is honestly definitely a community effort that involves people at all levels. And also thank you for the generous support from organizations that have provided resources to make our work possible. Thanks.
All right, thank you so much, Bryce. Oh my gosh, we have so many questions to get into. It's just striking the amount of plumage variation seems to be the name of the game here, right? Within species, within the subspecies, within individuals over the time of year. It's hard. It's interesting, for the non-scientist to understand how there can be such variability, but yet what defines a species really gets kind of it gets mixed up. So yeah, it was. So if you had to sort of summarize it, what if based on what you know today, the adaptive advantages to all this plumage variation, if you had to kind of put one sentence to that, how would you describe that?
The adaptive advantage to the plumage variation. So I'll just maybe frame the question in another way to make sure I'm understanding the question correctly. I mentioned that red-tailed hawk plumage generally varies across the landscape where we see changes in habitat, and that suggests that there is an adaptive advantage to being a certain phenotype in a certain landscape. And in raptors in general, a lot of that is probably related to their predator-prey dynamics, and their ability to for red-tailed hawks, they sit and wait mostly as they hunt. And if you're a predator that is sitting and waiting for an opportunity to catch its prey, you need to make sure that that prey doesn't immediately recognize you when it comes out of a burrow or something like that. That might impact. So over time, selection might select for more cryptic plumage in certain habitat types. Mm-hmm. So generally in open landscapes in red-tailed hawks, we see lighter birds because they're either hunting in the air or they're perched in areas where there's a lot of sunlight and not a lot of trees. So for instance, in the Great Plains, a lot of birds have really weak belly bands. And if you think about the subspecies crider, this very pallid, this very white bird, it occurs in the Great Plains where there's not a lot of tree cover, and a lot of sunlight. So that probably is related to their ability to match the backgrounds and not be easily detected by their prey. In other places like the Pacific Northwest, we see heavily marked birds with a lot of rich coloration that might help them blend into a more heavily forested landscape.
Mm-hmm. Yeah, that sounds great. So identification is a challenge with a lot of people, especially areas where the overlaps are closer. Where do you have any advice on how best to bone up on identification of these species, subspecies?
Yeah, no, it's great. Raptor ID is one of my favorite things. That's how I really got my start in raptor biology, and I love the game of having to identify raptors, whether it's through in the field or through photos. There aren't a lot of resources out there for understanding what name to put to certain redtails. But for those of you that are interested, that's what I'm trying to build on the website through the subspecies guide, is this really fully fleshed out resource for identifying each of the different subspecies, so that if you take a picture of a bird, you can go to that resource and use it to understand what subspecies you might have taken a photo of. There are other descriptions in all of the guidebooks that are kind of a surface level of the variation in red-tailed hawks that you can go to. Jerry Ligori published a few books that focus on raptors in flight, and particularly the distance, and he outlines how to tell certain subspecies apart. The Hawkwatch International has an in-hand guidebook that does a pretty good job of showing the differences between some of the subspecies. A lot of the resources out there for subspecies identification aren't comprehensive, and so you'll get a guidebook that's focused on the birds of North America, right? It's not going to have the birds it's not going to have a description on how to tell apart the subspecies in the Caribbean or in Mexico and Central America. So to do that, you would have to bolster your library and just look at a bunch of different sources. The Birds of the World account does have a pretty nice description of all the different subspecies, and you can use those descriptions and also leverage the Macaulay library to try and go to those descriptions, look at the Macaulay library, and see if you can understand what they're describing in words.
Mm-hmm. Yeah, and if you're lucky enough to live anywhere near a hawk count, going there and working with an expert side by side is always a great thing too, right?
Absolutely. In fact, that's a rule of mine for going to bird. If I want to be a better birder, I go birding with better birders than I am. And I was lucky to learn Hawk ID directly from Jerry Ligori, and that spurred my education in that rapidly. A lot of people don't have the luck of knowing somebody that's very, very talented or very knowledgeable in raptor ID, but there are a lot of knowledgeable people out there. Another resource for folks that want to start getting into it is the Facebook group Raptor ID. There's a lot of knowledgeable people that are answering questions about Raptor ID on that page, so that's a fun place to go if you want to learn.
Great. Yeah, our Birds of the World account is dedicated to Jerry, in fact. So.
Yep.
So yeah, lots of questions. So can you tell us a little bit about the size differential in the males and the females and the lifespan of a red-tailed hawk?
Yeah. Both things that we are currently gaining a better understanding on and hope to publish things about in the future on a comprehensive scale. Raptors in general are what's called sexually dimorphic in size. the males and the females are actually, the females are bigger than the males on average. In red tails, that is certainly the case. However, there's a lot of overlap. So when we catch a bird, we can generally get an idea of what sex it is by how much it weighs and how long the wings are and the tail are and how big the feet are and the bill, because females are generally larger. But that overlap is so much that a lot of people argue that there's too much overlap to be certain about the sex. So we have to use molecular tools to confirm what the sex is. However, nobody's really, a lot of the data that have looked into the differences in males versus females and described that overlap are from migratory individuals. So we're hoping to create a distribution-wide data set that shows whether some populations, some subspecies have bigger differences in male and female, and whether in certain regions of the species distribution, you actually can tell just by catching a bird and looking at its weight, whether or not you have a male or a female.
Right. If it's outside the zone of overlap, right?
Yeah.
Okay, so some people are talking about the similarities to the steppe buzzard or the common buzzard, Buteo, and somebody else was wondering about Harlan's similarity to Calaris subspecies.
Yep.
So do you have any thoughts on that?
Yeah, so I do. And for those people asking the question about Harlan, you're going to be very excited when we start publishing all the genomic work, because I already have some insight into that I'll unfortunately have to keep to myself for now, because we haven't really, it's still half baked, and I need to make sure that we explore things fully before I start sharing what we found. But Colourus in plumage, so Colourus has this really big range, and there's a lot of variability in the appearance of Colourus throughout the range. It's the western subspecies, so the description of where Colourus occurs is from parts of Arizona and California in the south, all the way north to central British Columbia. That in the north, they run into the population of Harlani as the southern distribution of the population of Harlani, and they actually there's plenty of documentation of them breeding with Harlani. So you get some Harlani that look a little bit more Colourus-like, but in general, Harlani is very different looking from Colourus. And in previous research that has looked at genetic data, they've shown that Harlani is actually not very similar to Colourus at all genetically. They're more similar to Borealis in populations in the eastern part of the continent. And that's something that that phylogeny that I showed actually supports as well, that they're most similar to criders in my sample set. And that's something that I can share from the sequences that we've gotten back and looked at so far on the comprehensive level, that Colourus is not very similar, is less similar to Harlani than the other red tails are to Harlani. So Abieticola and Borealis and criders are more similar to Harlani than they are to Colourus. I've forgotten the other part of the other aspects.
We asked about the common buzzard.
Oh, yes. Yeah.
Where's that range in that one?
Yeah. So Buteo diversity in general is very, very interesting. And there's a few papers that are fun to look at that describe that show the phylogeny or the relationships between all the different Buteos. The common buzzard, a lot of people like to say that the common buzzard is the ecological equivalent to our red-tailed hawk. It's a very, as the name implies, a very common Buteo that's found throughout Europe, and it is very similar to red-tailed hawk in being, with all the plumage variability it has. So it has a lot of subspecies, and it also has it's polymorphic as well. And there have been a lot of research on that. Surprisingly, though, it's not that closely related to the red-tailed hawk. So the red-tailed hawk, given what we understand now from the phylogeny of Buteos, the Buteos actually originated here in probably in Central America or South America, and radiated outwards across the land bridge into the Old World. And the youngest species are actually found in Asia and Africa. And the common buzzard is a part of that radiation. So as far as we understand, it's more similar to those species over there, of course, than it is to the red-tailed hawk. So the similarities we see are probably just driven by the similar selective pressures that they have acting on them.
Okay. Somebody wants to know if the dark morph could the dark morph have been selected for its temperature regulation advantages of more northern populations?
Yeah.
Sure.
Yeah, it's a cool question. So there are many different hypotheses to explain why we might see dark types in certain areas and not in others. I'm actually currently working on revisions on a publication that I hope will come out fairly soon that gives a nice overview of what we do and don't know about plumage polymorphisms in raptors in general. That'll hopefully provide some insights and some direction for research into understanding why we see it in the red-tailed hawk and why we don't see it in certain areas of its distribution. Things that might be at play in the red-tailed hawk for why we see dark types. Yeah, it could be related to temperature regulation. It could be related to ectoparasites and the amount of ectoparasites that certain populations have to deal with. So we do know that the amount of melanin in the feathers causes them to be stronger, provides some structure that makes them more resistant to ectoparasites that eat the feathers. It also makes them more resistant to abrasion and things. So in birds that are in more forested landscapes might be better off if they have darker plumage. There are also other things related to their foraging ecology. So in certain areas, it might be related to the suite of prey that they prey upon. Dark types might be better at catching certain prey types than light types and vice versa. And that balance between the two and their roles in pairs or at the nest might help them be better as a team. Because they can catch a wider diversity of prey, because certain prey types don't recognize the dark type. And also there's a lot of things on a mechanistic level or on a cellular level that might be at play. So certain parts of the development of melanin as the bird is growing a feather, some of the products of creating that melanin actually have a role in the physiology of the bird. And through what's called pleiotropy. So the gene controlling a certain aspect of the creating the dark type might also give it an advantage in that it's more resistant to parasites, or it might have a stronger immune system or immune response. It might be better able to handle stress under certain conditions. So what I think a lot of the research has shown, not to get too in the weeds, is that dark and light types in other species actually probably have a suite or a cocktail of traits that make one more advantageous in certain situations and the other type more advantageous in other situations. And so there's kind of this balance of pressures that create a situation where one is better at surviving than the other and vice versa. On a pretty restricted level within the population, creating the different plumage types.
Yeah, so speaking of populations, I think a lot of people want to know overall, is the red-tailed hawk population in general, how is it doing status-wise? And are you seeing more pressure on a certain subspecies than other? And then I would take that a step further and say, well, it covers so many wide variety of habitats. What do we know about the core needs? Like what does the core needs in terms of either habitat or food availability? Do we know anything about that?
Yeah, we don't, at least on a robust level. And that's something that I think our data can help with, especially our movement data. And I'm working with a Cornell undergrad to look at variation in home range size during the breeding and what factors might influence that variation. So, why do some birds have bigger or smaller home range than others? Is that related to the habitat that they're in? Are birds that are nesting in more human impacted landscapes, do they have larger home range because they have to go further to find food, or vice versa? So I think we can figure that out through our data. As far as the population status, because the species occurs in such a broad distribution, of course, there are going to be hotspots of declines and things for any number of reasons. Raptors in general, although red tails seem to do well alongside humans, they do face persecution. People are still out there shooting red tails. And they're also poisoning rodents that red tails eat. So there's rodenticide issues that are probably at play, things that we as a working group can work with other people to help us understand on a comprehensive level how big of an impact that's having. If it, on that status and trends map, you can actually see that there are areas, particular areas that have bigger circles and darker red, indicating that yes, there are certain spots where they are in decline. But another important aspect about that map is that some areas we just don't have the enough information to understand what the status of those populations are. Those are smaller populations in places like the tropics, and in places that are harder to get to. So the more eBird data we get, the better those status and trends maps will be, and the better we're going to be able to pinpoint those areas of concern for the species. And then we can focus our research on those areas and understand what aspects might be driving those declines and what is important for red tail, whether it's certain food or certain habitat types, to make it better, to bring those populations back.
And so one of the things that we continually learn is the importance of the Macaulay Library media archive. So when people not only report a red-tailed hawk, but if they provide a photograph or video of it, this helps you do this kind of research and really kind of tease out all of these questions, right?
Absolutely. I mean, like I was just referencing, eBird lists of, eBird complete checklists in underbirded areas. It's a great way to pinpoint, pushing the stats and trends map, the robustness of the stats and trends map. But also taking photos of birds in the Macaulay Library is incredible for me as a researcher, because what that is essentially data, right? There's so much information in every single photograph, things that I can't even think of right now, but maybe I will in 10 years. All of a sudden realize like, oh, that would be a really interesting thing to look at, and I can just go look through the Macaulay Library at photos and pull data like that. One example of that is our plumage aberration work. I'm working with a student, Amaya, and other students that were on that Oklahoma trip. Amaya's currently looking through Macaulay to catalog all the plumage aberrations that people have uploaded, so that we can gain a perspective of how many birds there are with plumage aberrations relative to how many red tail photos there are in general. To get it's an imperfect look, but it's a first step in understanding how common that trait is and what certain types there are. So yes, the Macaulay Library is and will continue to be a very powerful resource for the Red-tailed Hawk Project, but other researchers in general, for who knows how many things.
Yeah. All right. How can people help with your research?
Well, I think we have touched upon some of that, and that's participating in eBird, Macaulay Library, and things like that. Of course, I would always say money helps. It definitely helps. But more so, it's just, I think if people are interested in participating, it comes at all levels. That's sharing information that we share. That's, talking to people about red tails, getting people excited about red tails. On a more direct level, people interested in contributing can just reach out to me and talk to me about opportunities. We're trying to build a network of rehabilitators, of wildlife veterinarians, of all these people. So just having communication and getting in touch with people that are involved in our project, that's help as well, so that we gain a network of people that we can talk to across the continent, so that we can, if we have a question or if we need information about the birds in a certain area, we can tap that network and get that information. That's a huge help. So as simple as reaching out.
Sounds good. And that Facebook page again, what's it called?
It's just the Red-tailed Hawk Project on Facebook, and that's a great place to ask questions. You can share things and discuss things. That's a great forum for getting involved. But our website also has a contact us section that I do a fairly good job of monitoring and responding to as quickly as I can. So if you want to leverage that, that's a good way to get in contact with us.
Sounds good. All right. Well, this has been wonderful. I really appreciate you taking to the time, and everyone for being here today. If you have any more questions, certainly reach out to Bryce anytime. If you'd like to be part of that study, we really encourage you to. And if you can add any data to eBird, we really appreciate it. And if you can go the extra mile and add an image, that really helps the kind of research that Bryce does. So thank you all for coming, and we'll see you next month. We're going to be speaking with Elliot Miller, who's going to be talking about avian phylogeny. So it should be really interesting next month as well. Thank you all for coming, and have a great day. See you later, Bryce. Thank you so much.
Thank you.